Welcome geniuses, I'm Genie, your best buddy for A-levels. In this channel, we'll bring you to explore the secret formula behind success.
This video is on AS topic 5, the mitotic cell cycle, and I'm going to focus on the learning outcome 5.1a, which is about the structure of a chromosome, where you need to be able to describe the structure of chromosome during prophase or metaphase and anaphase, and I will introduce you with the different terms that you will call a DNA when they are at different stages. So let's go into the introduction. Okay, so there are a few names that the DNA can be known as. So the most common one that we know here is, during interphase, the DNA is actually loosely coiled DNA known as chromatin. So this is where you see double-stranded DNA associated with histone protein. So during interphase, what happens to the DNA? DNA will undergo DNA replication. So from one DNA molecule, you will be getting two identical DNA molecules. Two genetically identical DNA molecules. Okay, so during interphase, the DNA exists in a loosely coiled form. Then during prophase, that's where chromosome becomes visible under light microscope. So what makes them visible under light microscope? That's where the DNA will start to condense. So DNA will coil around its histone protein, forming chromosomes. So that's how you will see a ribbon-like chromosome under light microscope. So this is chromosome. So how would you describe the detail of this chromosome? So this particular chromosome is made up of two identical sister chromatids. So the DNA now is known as sister chromatids. So each of the DNA molecule is one of the sister. So you have two copies of the chromatids and held together by the centromere. So if I ask you to describe the structure of chromosome during prophase, this is how you imagine the structure of the chromosome, with additional structures such as, at the end of the chromosome, we know that they have these extra nucleotides that does not code for vital genetic information, but they are important to ensure the continuous mitosis and DNA replication. So this structure is known as telomere. For the understanding about the role of telomere, please read up on the importance of enzyme telomerase and telomere in stem cells and cancer cells. So this is how a chromosome looks like during prophase. Then how about metaphase? Okay, we know that metaphase is where the chromosomes line up at the equator. So they still look the same, So I'll just slash prophase and metaphase. Chromosomes looks alike. But during anaphase is where we observe that the chromatids starts to separate. So when the sister chromatid starts to separate, the centromere would have duplicated and divided. So now you will see that the sister chromatids has become a daughter chromosome. They are still known as chromosome because each of them has their own centromere. But now the difference is they are left with only one chromatid each. So the chromosomes will be pulled to the opposite poles, and this is known as anaphase. So when they reach telophase, what happens now? The chromosome will start to uncoil and they are going to go back to its original shape where it is known as chromatin again. So that's where the cells will be going through the last stage, which is cytokinesis, and they will enter interphase again, getting ready for the next round of mitosis. So let's relate to a sample question. Let's look at one sample past year question where it's from May June 2016, paper 22, question 5, where this question is actually expecting students to be able to tell how is the chromosome behaving during early prophase and subsequently becomes visible in late prophase. So the question gave an idea or the identify for you that this is the cell at the beginning of prophase. So what can you see here? Strands of chromosome, but it's not really obvious. And how does the late prophase look like? So they didn't provide a diagram here, a photomicrograph, but usually in practical, if you have come across, you'll see that if the chromosomes are condensed, you will see very obvious strands of chromosome. So what is happening to the chromosome that actually converts it from a thin strand to a thick strand? So here the expectation is just the chromosome starts to shorten and condense. So with this, the chromosome becomes visible under light microscope. So for the next question, how would you describe the structure of chromosome in late prophase? We know that the chromosomes are already in a condensed form. So that will be the answer for part 1. So you are not going to repeat the same answer again. So for this part, you have to relate to the structure. How does the chromosome look like? So we know that the chromosome will look like this. Remember my earlier explanation? It will be like this kind of like ribbon-like chromosome. But the idea here is why we don't see it in the photomicrograph. Because we have so many chromosomes within the cell, so it's unlikely that you will be able to see individual chromosomes. So we have to know theoretically that the chromosome will look like that. So how would you describe the structure? So number 1, you can say that the chromosomes now actually have 2 chromatids and they're identical. So 2 is one mark, sister chromatids are another mark. And then the next thing is, who is holding them? Another mark will be the centromere. And then another point will be, if you can think of, we know that at the end of the chromosome, there's actually telomere. So in multiple choice, if the question asks, how many telomere can you find in the chromosome during prophase? So here you can see that there are actually 4 telomere. But after anaphase, when the sister chromatid splits, where each chromosome only have 1 chromatid, then you will only find 2 telomeres. And one more point that if you can relate is like all the DNA, regardless whether you are in chromatin form or in chromosome form, you know that they are actually made up of double-stranded DNA coiling around histone protein. So this is how you can obtain the 3 marks for this question. The understanding of the different terminology to be used in different stage of the mitosis is very important, because during structure, you need to be able to use the correct term when you're describing the behavior, and especially for multiple choice, when they're using a wrong term, it shows that that particular statement is actually wrong. So hope you understand and able to differentiate all the different terms for the chromosome.
That's all for today's video. If you're interested in more genuine sharing by other geniuses, please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with 1 or 2 past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.